For decades, neuroscience has treated intelligence as an architectural problem. We've mapped the connectome, tracked synaptic plasticity, and modeled human cognition strictly as a function of how well our neural networks wire together. But the capacity for complex thought is heavily bound by raw cellular thermodynamics. Deep inside the cytoplasm of these neurons, an immense amount of energy is required to fire every signal. Look at this structural visualization. The human brain accounts for a mere 2% of total body mass, yet at rest, it consumes about 20% of our total energy output. Every perception, memory, and moment of focus you experience carries a steep biological price tag. Thought itself is a metabolically expensive act. Sustaining it requires a continuous, highly localized energy supply. Mitochondria function as dynamic regulators, moving beyond the role of simple cellular powerhouses. They constantly adjust to stress, modulate inflammation, and dictate our overall cognitive capacity. Intelligence is inseparable from metabolism. The clarity and resilience of a mind are shaped in equal measure by its networks of neurons and its networks of cellular energy. To see how these energy networks govern our thinking, we can look at molecular research conducted here at Imperial College London. The process that produces our cellular energy is called oxidative phosphorylation. Right at the start of this respiratory chain sits mitochondrial complex 1, or MCI. If this enzyme falters, the entire process of efficient energy production breaks down. Molecular psychiatrist Oliver Howes and his team brought in healthy human volunteers, measured their cognitive performance, and then used PET scanning to map the availability of MCI directly inside their brains. This visualization maps their results. The scans revealed a direct, positive correlation. Greater availability of the MCI energy enzyme mapped precisely to higher IQ scores. We see this same energetic bottleneck in animal models. At Mount Sinai in New York, researchers zoomed in on the physical architecture of synapses inside the prefrontal cortex of rhesus monkeys. Monkeys demonstrating strong working memory, the ability to hold and manipulate information, had synapses packed with dense, structurally intact mitochondria. Conversely, those with poor working memory had malformed structures. Their local energy grids showed clear signs of oxidative stress and reduced ATP production. Complex, sophisticated cognition requires a rigorously reliable energy system. When the localized structural reliability of the brain's bioenergetic grid degrades, intelligence is physically constrained. This biological efficiency scales far beyond individual synapses. We can trace it across entire human lifespans through data analyzed here at the University of Edinburgh. In 2017, researchers examined data from the 1947 Scottish Mental Survey. This was a massive cohort that tracked the childhood IQ scores of 70,000 individuals tested at age 11. This chart graphs survival to age 79. Childhood IQ is on the x-axis, chronological survival on the y-axis. The trend line is clear. Higher IQ strongly predicts a longer life. Counterintuitively, even mortality from cancer or accidents groups along this exact same statistical curve. Evolutionary biologist David Geary provides a mechanism. He proposes that high intelligence is an early, highly visible expression of a deeper biological variable, the systemic efficiency of the individual cellular energy. This energetic machinery follows a strict matrilineal script.
Mitochondria carry their own DNA, separate from our nuclear genome. At conception, the paternal mitochondria carried by the sperm are actively dismantled and destroyed, leaving only the mother's biological power plants. Because the brain is such an energetically voracious organ, tiny genetic variations in mitochondrial efficiency compound heavily. Over decades, a fractional advantage in ATP production expresses itself first as cognitive capacity in childhood, and eventually as physical resilience against disease. Biological aging, then, is a widening gap. The creeping deficit between the body's systemic demand for energy and the cellular machinery's ability to supply it. There is a cost to keeping that supply running. Oxidative phosphorylation generates ATP, but also produces a highly volatile byproduct, reactive oxygen species, or ROS. Look at this parameter curve. At moderate levels in this optimal corridor, ROS act as essential signaling molecules. But push production too hard, and the excess ROS begins stripping electrons, permanently damaging mitochondrial DNA. Researchers here at Columbia University are mapping exactly what pushes our cells out of that optimal corridor, connecting human psychology directly to hard cellular biology. Martin Picard introduced a concept called metaboception. He posits that the brain continuously monitors the entire body's energetic status, reading our emotional and psychological states as data inputs. When experiencing psychosocial stress like chronic anxiety or loneliness, the brain registers an acute threat, causing a systemic energy demand spike. To signal this deficit, the body releases GDF15. Elevated GDF15 suppresses the telomerase enzyme. Without repair, protective chromosomal telomerase rapidly erode, accelerating biological aging. Chronic loneliness and psychological stress manifest as physical, measurable metabolic events that erode the very architecture of the cell. Optimizing cognitive performance requires treating the brain as a strict energy budget, one that requires deliberate management and repair. Physical exercise is a powerful metabolic lever. Intense physical exertion triggers a process called mitogenesis, prompting your cells to dynamically build completely new mitochondria, permanently expanding your baseline energetic capacity. That expanded capacity demands maintenance. Consistent circadian sleep alignment is a biological necessity because it enables mitophagy, the required systematic clearing of burned-out mitochondrial debris and accumulated oxidative damage. You also have to provide the correct raw materials. Targeted nutrient sufficiency, specifically B vitamins, magnesium, and unsaturated fats, gives your cells the precise molecular components needed to stabilize the oxidative phosphorylation pathway. Finally, building social connection and intentionally mitigating psychosocial stress acts as a direct metabolic intervention. It limits the release of GDF15, lowers your cellular energetic tax, and preserves the length of your telomeres. 2 billion years ago, an ancestral cell engulfed a smaller bacterium and, instead of consuming it, merged with it. Our entire capacity for thought traces back to that ancient merger, an alliance founded on cooperation rather than domination. We carry millions of billions of these ancient collaborators inside us. A brain capable of navigating the complexities of the 21st century must first understand and actively respect the bioenergetic foundations that make thought possible.